Bismillahirrahmanirrahim. Dear students, have a nice day. Hope you will be fine and best of your health. This is Dr. Sajid Mahmood Rao, Division of Science and Technology, University of Education, Lahore. Today we are talking about short question answers, which is relevant to the Advanced Natural Product Chemistry, Chem 5121. Let's see the short question number 8. Define hormones. How dihydrotestosterone has a more potent androgenic effect than testosterone. Let's see what are the hormones and dihydrotestosterone is derived. Androgens are one of the main families of steroid hormones present in humans. Progesterones, androgens, estrogens, glucocorticoids, and mineralocorticoids. The androgens group consists of four hormones, that is dihydrotestosterone, testosterone, androstenedione, and dihydroepiandrosterone. The relative potencies of these androgens are 300%, 100%, 10%, and 5% respectively. The principal androgens are testosterone and androstenedione. They are of course present in much higher levels in men and play an important role in male traits and reproductive activity. Other androgens include dihydrotestosterone, that is abbreviated as DHT, dihydroepiandrosterone, that is abbreviated as DHEA, and dihydroepiandrosterone sulfate, that is abbreviated as DHEAS. The androgen receptor is a member of the nuclear receptor superfamily and binds both testosterone and dihydrotestosterone. These receptors reside in the cell cytoplasm bound to heat shock proteins until testosterone or dihydrotestosterone diffuse into the cell and bind the receptor, resulting in dissociations of the heat shock protein. DHT has a more potent androgenic effect than testosterone because its binding affinity to the androgen receptor is approximately double to that of testosterone, and moreover it has a dissociation rate about a fifth of that of testosterone. Let's see in the diagram how the dihydrotestosterone is derived from the concerned hormones. As you know, and you can see also over here, the testosterone is derived from the cholesterol. The difference between both the structure is that there is a presence of hydroxyl group at carbon 3 positions and a side carbon chain at 17 positions and a double bond at carbon 5 position in the cholesterol, while that of in testosterone the carbonyl group is present at carbon number 3 and hydroxyl group is present at carbon number 17. So in this way, from testosterone, the dihydrotestosterone is derived by the effect of enzyme 5-alpha reductase, and estradiol is also derived by the enzyme effect, that is P450 aromatase. In this way, the dihydrotestosterone is derived from the first intermediate, which is synthesized from the cholesterol. Let's see the short question number 9. Discuss the structure and function of dihydrotestosterone. Explain how dihydrotestosterone is biologically important for sexual differentiations of the male genitalia during embryogenesis. Now come to the real topic, that is dihydrotestosterone. Dihydrotestosterone, that is also known as DHT, 5-alpha dihydrotestosterone, 5-alpha DHT, androstenolone or stenolone, is an endogenous androgen sex steroid and hormones. DHT is one of four principal androgens in humans and is synthesized primarily via the irreversible action of microsomal SRD5A, both types 1 and 2, on testosterone. DHT is the 5-alpha reduced metabolite of testosterone that is principally converted from testosterone in target organs such as prostate, skin, and liver. Synthesis can also occur from other precursors, but these pathways, although potentially important in some tissues. The common example is prostate, which is minor. Intracellular DHT is a more potent androgenic agonist than testosterone, and its presence in some tissues such as prostate is necessary for the full organ development and function. Circulating dihydrotestosterone levels are of much less important than testosterone for optimizing the intracellular DHT concentrations due to the presence of a rate-limiting enzyme, that is 5-alpha reductase, and is shown in SRD5A in types 1 and 2. Inhibition of these enzymes with 5-alpha reductase inhibitors decreases intertissues in dihydrotestosterone levels, and thus in certain tissues, that is prostate, that diminishes the agonist action of testosterone, thus reducing prostate size and function. You can see over here the basic skeleton of dihydrotestosterone, in which total number of carbon atoms are present 17, and the carbonyl group at carbon 3 position and hydroxyl group at carbon 17 position. Dihydrotestosterone is biologically important for sexual differentiation of the male genitalia during embryogenesis, maturations of the penis and scrotum at puberty, growth of facial body and pubic hair, and development and maintenance of the prostate gland and seminal vesicles. It is produced from the less potent testosterone by the enzyme 5-alpha reductase in selected tissues and is the primary androgens in the genitals, prostate glands, seminal vesicles, skins, and hair follicles. DHT signals mainly in an intracrine and paracrine manner in the tissues in which it is produced, playing only a minor role, if any, as a circulating endocrine hormones. Localizations of SRD5As in prostate tissues, that is type 2, skin type 1, liver type 1 and 2, and hair follicles, that is primarily type 1, catalyzes the formations of dihydrotestosterone from testosterone in these tissues. These enzymes expressed in the nucleus and cytoplasm of, for example, prostate epithelial cells are encoded by the 5-alpha reductase type 2, that is SRD5A2 gene, and polymorphism of this gene, leading to increased 5-alpha reductase activity and dihydrotestosterone concentrations in the prostate, that is have been hypothesized to increase risk of prostate cancer. The SRD5A3 gene has also been linked to increased dihydrotestosterone production in hormones refractory prostate cancer cells, and these genes may be particularly important in the metastatic prostate cells, which have been shown to express more SRD5A1 and SRD5A3 but significantly less SRD5A2. Conversion of testosterone to dihydrotestosterone via SRD5A activity in peripheral tissues is the main source of circulating DHT, but it is important to note that little DHT synthesized in the prostate or liver enters the general circulations due to efficient intracellular mechanisms that initially metabolize DHT to 3-alpha and 3-beta 17-beta androstenedione diol that have little androgens activity. As noted previously, DHT can also be synthesized in tissues by backdoor pathways that enable formations of DHT in the absence of testosterone or androstenedione as precursor. In yet a third synthetic pathway to dihydrotestosterone, namely the 5-alpha androstenedione pathway, 5-alpha androstenedione is converted by 17-beta hydroxysteroid B3 to DHT. As discussed later, these alternate synthetic pathways, which are not influenced by circulating dihydrotestosterone, may have particular clinical significance within prostate tissues. There are two isoforms of the 5-alpha reductase enzyme. Both are microsomal NADPH dependent proteins that reduce the double bond at the carbon 4 to 5 positions in a variety of carbon 19 and carbon 21 steroids base skeleton. They differ in regard to their optimum pH, chromosomes locations, and affinity for testosterone. Let's see the short question number 10. Explain schematic effect of 5-alpha reductase 1 and 2 enzyme on testosterone. How steroidal hormones are derived from dihydrotestosterone. So you can see over here how the dihydrotestosterone is derived from testosterone by the effect of enzyme 5-alpha reductase 1 and 2, that is also abbreviated as SRD5A1, 2 and 3. This is the first step which is used for the synthesis of dihydrotestosterone. Let's see how different kinds of steroidal hormones are derived from dihydrotestosterone by the effect of relative enzymes. You can see over here in the second step 3-beta 17-beta androstenedione, which is a steroid which is derived from dihydrotestosterone by the effect HSD7, that is 3-beta DH. Similarly, the second steroidal hormones, that is 3-alpha 17-beta androstenedione, is derived by the effect of enzyme HSD5, 7 and 10, that is also known as 3-alpha DH. Both these enzymes are isomers of each others because there is a difference only the positions of above and below the planes of 3-beta and 3-alpha of these steroidal hormones. After that, 3-alpha 17-beta androstenedione gives 3-alpha androstenedione glucuronide by the effect of enzyme UGT2, B7, B15 and B17, and also gives androstenedione by the effect of enzyme HSD2, 5 and 11. This enzyme finally gives androstenedione glucuronide by the effect of enzyme UGT2, B7, B15 and B17. The dihydrotestosterone also gives androstenedione by the effect of enzyme HSD2, 5, 8 and 10. Further, dihydrotestosterone gives DHT glucuronide by the effect of UGT2, B7, B15 and B17. Finally, the steroid 3-beta 17-beta androstenedione gives 3-beta androstenedione glucuronide by the effect of enzyme UGT2, B7 and B15. This is the basic diagrams and schematic pathway for the synthesis of dihydrotestosterone. Type 1 5-alpha reductase has an alkaline optimum pH of 8 and is active over a broad pH range, that is 6.0 to 8.5. The gene encoding type 1 5-alpha reductase, that is SRD5A1, is located on chromosomes 5 at PL 5.5, while that encoding the type 2 enzyme, that is abbreviated as SRD5A2, is located on chromosomes 2 at 2p23. The type 2 5-alpha reductase has at 10 to 15 fold higher affinity for testosterone than the type 1 enzyme. The type 2 5-alpha reductase has an acidic optimum pH of 5.5 and in contrast to the type 1 isoforms is only active over an extremely narrow pH range. The two enzymes are differentially expressed and regulated in the regard to tissues distributions and timing of expression throughout the life. The ontogeny of the 5 alpha reductase isoforms has been elegantly clarified by the author Thaifen et al., who have demonstrated the dynamic expressions in humans. The type 1 enzyme is not detectable in the fetus, appears transiently in newborn skin and scalp, and is permanently expressed in the skin, including pubic skin, only after puberty. There is no difference in type 1 expression between bleeding and non-bleeding scalp. The type 2 enzymes has been found to be transiently expressed in the newborn skin and scalp, the adult scalp, and pubic skin. Within the male reproductive organs, the type 2 enzymes has been localized to the testes, epididymis, and vas deferens, while type 1 appears more abundant in the seminal vesicles and ventral prostate. Let's see the comparative study of type 1 and type 2 5-alpha reductase isoenzymes, which is located in the tissues. In type 1, we can see over here it is transiently present in skin after birth, while type 2 skin transiently after birth. So in type 1 skin permanently after puberty, in type 2 pubic and scalp skin permanently after puberty. In type 1 axillary hair and follicles, in type 2 beard hairs and follicles. In type 1, it is present in liver, seminal vesicles, and ventral prostate, while type 2 is present in internal and external male genitalia and liver. The liver is the main source of circulating DHT in both genders, as it contains both type 1 and type 2 isoforms of 5-alpha reductase. The DHT found circulating in blood is predominantly from type 2 5-alpha reductase activity, that is 70 to 80%, with the remaining 20 to 30% from type 1 5-alpha reductase.